All right, so here's the 3-minute version.
Wait.
Replication is going to be the complete copying of a set of genetic information. Replication is going to be the complete copying of a set of genetic information. All right. It occurs when you grow new cells, which happens basically 2 times. So when you are injured and you are repairing, or you are growing. Okay, so replication is the copying of DNA. The mitochondria must do it, and your nucleus must do it. In prokaryotes, the genomic DNA must do it. It is an exact copying, but sort of, because it has mistakes. We're going to differentiate this from what happens when you produce sperm or egg, right? So we are producing clones. We are not producing children here. So if I were to run replication, I would have a complete, exact clone of you. It would not be like I had your child, right? Not that I would have your child, because that would be all kinds of cool new science. All right, so it's these mistakes in copying that allow for all branches in kind of all trees. So, for example, at this split...
Both.
What?
Evolutionary
Both, yes, both. But evolutionary trees, right? So, mutations can be good or bad, depending on what they do for the organism. Actually, we might as well go ahead and say those mistakes might have, they generally have 3 effects. They make a protein work better. Oh, I don't like that anymore. That understanding has changed.
Well, it's on the page, so...
Can we make a product of DNA work better?
Wait,
So it could be an RNA. There could be no change, and there could be worser.
Oh, it means the same thing. Like make a product and then, you know.
Can I do that in my English essay?
Yeah. You should just do that in your English essay. Totally.
That's a bad idea.
Actually, you should take out all pronouns from your English essay and write like you do in science.
Oh God, it's like in Cynthia's class. No verbs of to be. Look, have you ever tried to rewrite 5 essays without the word to be?
That's an interesting.
Oh,
No, it's not.
Wait. Okay, we're getting off track. Okay, so this is accomplished in eukaryotes through a machine called, let's look at bacterial. So we'll look at bacterial replication. It's a little bit easier, but not at all easy. So if you think about it, replication must occur, and everything that happened in vitro in replication needs to happen in a organized fashion in the cell. So there's 2 machines, basically, that work together. One's called the replosome that does the actual copying, and one is called topoisomerase. I need a piece of string.
Take cord. Can you rewind?
I need a piece of string.
Just use someone's arm.
Where is my little tuber?
What was the second one? Topo.
I have no idea. I'm just gonna call it Topo.
Okay.
Good. They're about the same.
Topo.
Wait,
wasn't this simple?
Look.
I'm gonna rewatch this video, and I'm gonna listen for the part where I like laugh really loudly.
The variability from 22 to 24, and not from 22 to 24.
We should be like,
Hey, Luke's YouTube channel.
oh yes.
No. No, I bet Luke is wrong. It's not 26 in total. It's 24 from the mitochondria plus 2.
But I've seen it written before that there's 28 total, 2 from the cell.
Well, you know what, Luke?
Guys, this is our chance to talk on his video.
Wait, I understand. Can we please do that? Waste a little bit. Okay, Luke, Professor.
So
Professor.
Luke, tengo una pregunta.
this is DNA, and if you'll hold the side of this.
Yeah, Dylan.
What's going to happen is basically a replosome is going to identify a piece of DNA, a segment of DNA, called the origin. Right? So the origin is a sequence of DNA. It's like C, 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 T, C, A, G, C. I don't know what it is. Okay? And it's going to bind to this piece of DNA, and then it's going to start to open it. And one thing that you'll notice, hold tight, is that as it opens, the ends are going to start to coil tighter. Okay? So what will have to happen is an enzyme will come in, it will cut the DNA, loosen the coil, and then reattach it. Okay?
Seems like a really inefficient process.
Wait, what binds to the origin?
Because it seems like there'd be better ways to unfold it.
Yeah, talk to God.
What's it called again?
Okay.
What's the other one?
Okay, so
It's number I have been canceled.
step one in this process
Mold and like
is going to be recognition
Ready.
of the origin of replication.
What's the second one called? One that cuts?
Get it?
What is the thing that cuts the DNA?
Step one, recognition of the origin of replication. The site on DNA, the sequence, the set of letters, that is the origin of replication. That is where replication will start. Your DNA has many of these. And we put one in our plasmid. Do you remember what we called it?
Ori.
The Ori.
Can I write Ori or do I have to write Ori?
Okay. So, we said one of the first pieces of the origin of the replication complex will assemble on the Ori. So the thing that is doing the replicating is called the replosome. It has many parts. So piece of replosome.
And that binds?
And yep. The other thing that's going to happen is down here somewhere. Topoisomerase, which is a what kind of molecule?
Enzyme.
Produced by what?
Enzyme makers.
What makes enzymes?
Ribosome. RNA and ribosome.
Ribosomes make. Using information on what molecule?
The DNA.
Okay. Alright, so topoisomerase is going to cut the DNA and relieve tension. This is important. Without topoisomerase, your DNA would break as it was copied. Yeah.
So is that at a specific place too, where it cuts?
I don't know. I don't know how that works. That's a very good question. Okay. So, the replosome is composed of many pieces, and if I tried to draw it for you right now, your brain would break.
Do it.
No. Like, literally, you would all start crying. Okay.
Too late.
So, let's think of in vitro replication and think about all the functions that happened, and then we're going to find the in vivo equivalent. So in vitro replication we called what?
PCR.
Yes.
Wait, is this all step 1?
No.
Wait, what is vitro?
In like
Okay.
a test tube.
So step 2 is everything else that happens.
Oh, okay.
And it all kinds of happens at the same time.
So step 1 is like, oh, there's the thing, and then step 2 is like everything.
Yeah, it's like, okay, we're going to assemble on the replosome. We're going to assemble the replosome now that we have piece 1 there. And then it's like, everybody go. Okay? So, in vitro, we have heat.
And then sometimes there's an organized medium.
Now, heat melted
That could just help manage it.
DNA.
The cells go.
What was the point of melting the DNA? Why did we apply heat?
So it can
Go ahead.
denature.
Okay, so that it could denature. Function, strands came apart. Actually, why don't we try this?
Well, it's not because we died.
Okay, copy this down.
Oh.
Is this homework?
Oh, okay.
And tell me what the function of those next two things were. Talk in your groups. Talk. Talk.
Well, calling IDT DNA.
What does that mean? I literally don't
Okay, so like in vitro replication, like we called IDT DNA, so it was the function of that. Like what was the
What's
call?
Like, I don't understand what those three words mean together. Calling IDT DNA.
Remember, we got the plasmids from IDT DNA,
What's the one below?
What's IDT?
right? IDT
Oh,
is a
integrated
company.
DNA
It's a
technologies.
company.
Oh.
Oh.
Initialize.
What a calling.
Why
It's a
did we call
company.
them? Yeah.
Remember, we got the DNA.
No,
from
like
them.
calling the
We got the
calling the
plasmids
IDT
from
DNA,
IDT
calling the
DNA.
company. What,
I
I don't understand.
Luke,
What that
wait,
was
what
three
do you mean by
words
calling
together.
IDT DNA?
Why did I call IDT DNA, Mickey?
I have no
To get
idea.
the plasmids.
To get angry?
Wait,
That's
did
what I
you
would
get
do.
the plasmids?
You mean the good people
Nope.
at?
Yes, I called the good people at IDTNA for one specific thing.
DNTP?
Why don't you
For
just
the
look up
primers.
DNTP? Q5? Oh.
Why don't you just look up IDTDNA?
I
Because
did.
we
It's
did.
called Integrated DNA Technologies. It's based in Coralville, Iowa. It's the largest supplier of custom nucleic acids, serving the areas of academic research.
Right. They're primary
Yeah.
primary business.
DNA that charged 85 cents per nucleotide.
I love how you remember. I love what you remembered from that. Okay, all right. So, calling IDTDNA. What was the point there? Primers. Okay. And why do I need a primer? What do I need? What molecularly do I need? Toby.
The 3' end.
I need a 3' end of DNA. Okay, and adding tack. What was tack gonna do? What did it do?
It added, it elongated the primer.
Okay, sure.
Exponentially, right?
I mean, it added it one at a time. The result
then
was
it-
exponential.
Pranked.
He first added 1 on, and then just added 2 on, then 4 on.
What?
Okay.
Give me free money.
If you get a margin account.
So, can I heat your body to 90 degrees centigrade?
Yeah, you definitely.
We'll die, but
Alright.
whatever.
What would happen if I heated your body to 90 degrees centigrade?
I'd get really sad.
Like during the process, we would be sad, and then suddenly would not be sad anymore.
Very, very good. Alright, you would be sad and then not sad anymore. Okay, so we're actually going to use a molecule called helicase, which often is called MCM. Helicase is what you'll see most commonly in regular nomenclature, and you might see MCM in more advanced nomenclature like a paper.
Wait.
Okay. Helicase looks kind of like a donut.
Like a Taurus.
I'm pretty sure MGM is the much more famous of.
Hours.
There is a MCM, but this doesn't
So
look like
helicase, folks, is going to pull these two strands apart. Okay? Helicase is going to pull those two strands apart. Another enzyme is going to build my primer. It's got a nickname. I'm going to tell you the nickname. And then I'm going to tell you what it really is. I need you to remember both. Okay, so it is called primase.
Is that the nickname or
The nickname. But it is really an RNA polymerase. So in vivo, our primer is actually made of RNA. And it kind of, again, supports the idea that there's an RNA world. That before Phil, who used DNA to store information, there was an only RNA world. Everything was like RNA.
But like,
ATP was RNA, the energy molecule was RNA.
wouldn't all these like single RNA strands like want to bind?
Yes.
That's what happened.
How would they not? How would they just stay RNA all the time?
Because they're stable once a certain number bind together.
What?
Dylan,
Alright,
double strange RNA.
okay, so...
Yeah.
Yes? Question?
No, nothing.
Oh. Alright, so this is the most difficult piece. Our DNA polymerase, which was from the organism Thermus aquaticus, actually not from the organism Thermus aquaticus. Oh, I was wrong, Mickey was right.
What's it from?
Not from Thermus aquaticus, which is what matters. Alright, fine.
Okay, now this is a really confusing part. Our DNA polymerase is going to be from whatever organism we're in.
Relating back to the lesson.
So,
Wait, so
if we're in a human,
Pretty cool.
our DNA polymerase is going to be human DNA polymerase.
I don't get it.
But if we're in a leopard or a narwhal, actually let's use narwhal. Can anybody guess the type of DNA polymerase that's in a narwhal?
Narwhal DNA.
It is in fact narwhal DNA polymerase. In Spanish, El Narwhal DNA polymerase. So, okay.
Oh my gosh.
Oh my god, I forgot how awesome water bears are.
Alright,
Water bears.
okay, DNA polymerase is going to actually add the nucleotides. The problem is going to be that I'm now going to have a RNA-DNA combination. Does everybody understand this? So if I have a strand of DNA, I add a piece of RNA to it for my 3' end, and then I make DNA off of that. Does everybody see that I have a RNA DNA hybrid?
Yeah. Mm-hmm.
It might help next
There's
time if you
nothing
don't use
inherently
2
problematic
pinks.
This is
about
what I
that,
have.
right?
I'm working with what I have, Mickey. Alright? It costs $87 million a year to go to school here, and this is what they give me.
Oh crap.
Wait, what?
True.
It's, yeah, not wrong.
87.
No,
Okay.
no, not really. I mean, really.
No, it's actually more like
Let
90.
me tell you the part that you're going to remember, and the part that you're never going to forget. This fragment was actually discovered by Luigi Okazaki.
No.
His real first name is not Luigi.
Oh my god. Well, now I'm not gonna forget that.
But
I'm not gonna remember the last name. It's
his
gonna be
last
Luigi.
name was Okazaki.
What was his first name? Luigi.
I remember it has an R or an L in it.
Mario.
There was like a Japanese person, and they were teaching me how to say it, and it was like pronounced like a D, and that's all I remember.
It's closer to like 30
Okay,
million a
so-
year, right? When you Italian
When you Italian glaze?
when you
We have an Okazaki fragment in vivo, and so this RNA will have to be replaced.
Oh, that's pretty cool.
Seems like it'd be the most prone step for error, though.
Another DNA polymerase.
DNA trick.
Right? Like,
Wait.
Wait, I said actually Nueva has like from tuition.
Would you have to? That's a good question. I don't know the answer to that.
those are 20.
It is.
They were able to, if they wanted to, pay off the entire
Yeah,
cost of the
right.
building in a year.
Okay.
Raging
Of upper
Locust.
school.
Right.
22 million.
Yeah. That's it.
They
Oh my
cost 96 million.
Sure, that's including Baymeadow's license.
That is my least
Right.
favorite alarm in the
Building
whole
license.
world.
Oh yeah,
$22 million.
Alright.
but they paid it off the entire 96 in 2 years.
Yeah,
Wait, I'm sorry, Luke, what is the Okazaki?
honest to God,
it's
absurd.
So he discovered that it's
Like-
RNA and DNA.
Have you seen like the tree at the middle school with the like 2 million plus and it's just
Yeah.
Doing it.
Okay. So this is basically it. Of these, I would say there are 3 really important ideas.
Wait,
The first is that replication is going to happen at the origin, which is a sequence of DNA.
Is it Phil?
The second is that we're going to try to make an exact copy. And the third is that there will be mistakes. Yes.
What's the like approximate rate of mistakes in replication? In
I
vivo?
will go over that. There's each of the enzymes has a different rate of mistakes.
Specifically for the replacing of the RNA, like
There's a separate DNA polymerase specifically for the RNA.
right. But like, what do you know what the mistake rate is for that?
That one I don't know actually. I know the overall mistake rate is about 1 in a million.
per
Oh,
base pair
that's-
or per
Per million
entire
bases, there's one mistake.
Compare that to
Okay,
that'd be one.
so can we jump on Socrative?
That's really cool.
Cool. Yeah, just pretty.
Oh,
We haven't done soccer in so long.
True.
Sock broth.
Don't start yet.
Too late now.
Oh, I can just airdrop it.
When M goes to mine,
Wait, Luke, have you ever noticed that Luke D Bio 1 also kind of looks like Luke D B101?
I'm just gonna send it to
I have never
I'm not
noticed
speaking to you
that.
anymore.
It's because he's too blown away. He's like, oh my god, Mickey's right, like usual. Reminds me of this one time.
Luke dead.
Luke, why'd you send me an
This
email?
one time, right?
Oh,
When there was some like
what is
I see.
differences
Thank you for your
in
help on
ideas.
Friday.
Over
Oh,
Luke Devil
'cause
101.
I forgot your last name,
Denver.
and I thought you
Wait,
were
is
the other
this
Great.
actually
No, it's Luke D
it's
Bio 1, but it looks like Luke D B101. Isn't that a prank? I thought I couldn't get into the classroom the first few classes.
Like in this room, and like
They changed the font too, so it's a lot more clear. When we started, it was like a very non-serif font.
I see.
I have yet to answer something.
Wait,
other
what's
than...
my student ID? I think it's like 7.
No, 1 something.
Yeah, you're like 107, I think. Because I put yours in by accident. I was like, are you a real
Oh,
Johnson?
up top of room. We teamwork that.
Ouch.
Agree to disagree.
We start.
What did I learn in class today?
What would happen?
Nothing.
Actually, you know what? I have no idea what that piece of replosome is. I'm sure that I'm mistaking something about the replosome.
Wait, what do you mean? Which part? Wait, which part?
Wait, the piece of replosome? Just the piece of
it's-
replosome.
it's-
It's just
It's just a piece of
fuck.
I wish I could say that I learned about like what happens between the mitochondria and the rest of the cell, but I'm more confused than when I came in. There's no concrete number.
If topoisomerase were not used in
So it's the
Evolution would adapt and find a new way to relieve tension.
it's what opens the DNA. What does the replosome do?
So helicase is part of the replosome.
What did you learn today?
Folks, good question from Elise. The helicase is part of the replosome.
Nemo.
Nemo.
Okay.
Nemo.
This is why Nemo's fin was damaged. He didn't use
Yes.
it.
So the replosome contains all of these things.
Oh boy. Then what's the Topios?
Oh, that's why our head would explode.
So PCR.
How many
is
more
a
pieces are there than we see
No.
here?
Multiple,
Where is PCR happening?
maybe.
It was just one
In the
sample
or-
piece. It's just really, really big.
In the PCR thermocycling
Okay.
machine.
in vitro.
So, here, you want me to draw what a replosome would actually look like?
I'm funny. I know it.
No. You said we would cry if you did it.
Yes, I'm ready.
I'm already starting.
Challenge accepted.
Oh yeah, that's pretty bad.
What is that? You
No,
want to
wait,
try?
it's guys. It's just like 7 donuts.
Yeah. Come on.
Okay. Alright, so folks, the idea, one of the important ideas is everything here is together, right? So,
Oh, it's a snowman.
the helicase, the RNA polymerase, the DNA polymerase that is going to make nucleotides, and the DNA polymerase that's going to replace the RNA from the Okazaki, all together in the replosome. All together.
So
How quickly does the entire process?
All of these things make up the replosome.
Okay.
Our makeup.
We're a Spanish teacher.
Hey, Luco.
L.
Luco.
Yes.
Tengo una pregunta.
Okay.
¿Esmuéranse o muéranse? What? ¿Muéranse or muéranse?
Thank you.
It's an S
Yeah, for sure.
or muéranse.
Bunch of stuff like evolution of Phil into eukaryotes, process of ATP
Alright.
creation, and
So,
whatever.
what are the 3 big ideas? What are the 3 big ideas in replication? I want everybody to say the first one right now. Go.
Replication happens at the ori.
Mitochondria is the powerhouse of the cell!
It's what makes it
Alright, so one, what is it? What is the most important idea?
Replication happens at the ori.
Copying of a set of genetic information.
You can just assume that they said it right.
Replication occurs at the...
At the ori.
Okay, it's an origin. 2,
It's trying
It
to
tries
make
to make an
exact
exact
copy, but it fails sometimes.
it's an exact copy.
That has mistakes sometimes.
3,
It's
It
not
has
an
mistakes
exact
sometimes.
copy.
it's not an exact copy. There are mistakes.
It tries so hard. Yeah.
Okay?
What would happen just now?
Oh, wait, what was the teacher's question? Oh, that's cool.
What?
You know.
So,
Oh, it was literally
folks,
just
the
what
teacher's
you said.
question
No,
refers
no, the
to
teacher's
a bunch
question is
of
about
cancer.
topoisomerase.
And
I like the
if
questions.
you remind me, I will do a lunch with Brian on topoisomerase and cancer therapies. It's kind of cool. Starting next class, we're gonna actually start to be able to like understand real stuff now.
Now we're gonna start to be able to understand.
You just you're a replosome.
Ooh.
Oh my god.
Ooh.
You're a cow. What? It's a reference to us. So you can tell it. You're a cow. You're a cow. You're a cow.
Can I leave?